Hello and welcome to week 4. This week's content deals with the Human Genome Project and the ethical, legal, and professional dimensions that have arisen since the mapping of the human genome. Okay, so the Human Genome Project in 1990, there was a group of international scholars and scientists who wanted to identify the bases along the entire human genome and sort of make a map to determine the actual number of genes that humans have and find locations for important genes. The problem was that there are about 3 billion bases in human DNA, and they have to be in precise order, so mapping this out was quite an accomplishment. The equipment was fairly basic. It could only read about 1,000 bases at a time, so researchers broke the strand into smaller parts and then reassembled them like a giant jigsaw puzzle. And remember that when you break apart a strand of DNA, the sequence of bases on one side tells you about the sequence of bases on the other side. Some sequences were very complex. A lot of them are very repetitive. So it was a little bit of a puzzle for researchers to figure out what was actually going on with the human genome. But in 2003, the team announced the sequencing of the human genome was complete. It wasn't really. There were still about 400 series of bases, little pieces of DNA, that were either very complicated or very repetitive. These were thought to be junk DNA parts, DNA that really didn't do much, didn't code for important proteins. But they kept working on it, and March 31st of 2022, they finally had the entire sequence of bases in human DNA. It turns out there's only about 25,000 genes in the human genome, and it's not as complex as some species like water fleas or rice plants, and I think there are some trees that have like 100,000 genes. So that's fairly interesting information. I think they were a little disappointed to find out that the human genome was actually not as complex as they thought. But they were able to get some information from mapping the genome. When they knew where genes were, they could examine where the differences among humans with different traits. For example, if you study the Ashkenazi Jewish community, and the Ashkenazi Jewish community is unique in that there was a population bottleneck and they intermarried with one another, so there's not a lot of genetic diversity. But they studied people who lived to be 100 or more and people who did not, centenarians. And they found a sequence of DNA where 2 bases were much more common than in people who did not live to be 100. So the individuals who had like 2 cytosine nucleic acids in their sequence lived to be 100 plus at much greater rates than those who had CT or TT in that same gene locus. Differences in those single nucleotide pairs can be analyzed to identify the genes for many illnesses. It's easier to do it with single gene traits than with diseases that have complex polygenic causes. For example, cystic fibrosis, they were able to locate the genes that regulate that production of the protein that forms the chloride ion pump that's defective. Traits like diabetes, autism, obesity, very complex genetic sequences, it's a little bit more difficult to do that, although they've been able to isolate some of them. Human Genome Project led to the publication of the human genome on the internet. It also gave rise to testing for genetic disorders, and the newborn screening inborn errors of metabolism got much more refined. There was a creation of a catalog of mutations related to cancer, which is kind of exciting, that there are some genes that are related to cancer and tumor production and tumor suppression. So they were able to create a catalog of those mutations. They developed cheaper methods of sampling and analyzing DNA, therapeutics that are tailored to individual genome factors. And we'll talk about pharmacogenomics later on in this course. But it's exciting to see the development of treatments that are tailored to a person's unique genome. And they studied ancient humans and other human-like species to understand mutations involved in human evolution. So it was a very useful application. There are research priorities that the National Human Genome Research Institute has declared in their agenda. You can read about them on their website as well. Cancer research, When they started studying the human genome, so epigenetics is the influence of genes working with each other and with the environment. There are little switches that go on and off that can regulate certain functions in the body. So it's not strictly just having the gene. It's having the gene and then having that environmental trigger, or having a group of genes and environmental trigger. So epigenetics is a big priority for research so that we can understand more about how genetics raises our risk for certain diseases. Behavioral health, this is a very high priority for the NIH. Genomic epidemiology and population health, studying populations. They have small molecule therapies for genetic disorders, complex polygenic disorders such as diabetes, rare genetic disorders, inborn errors of metabolism. They are studying de novo mutations, those very first time mutations that occur in the germ cell of an individual, get passed on through generations, and the role of the microbiome and genetic variants of microbial organisms. So these are their research priorities, and there's a lot of interest in learning how genomics influences a person's health or population health. Now, CRISPR. CRISPR is a very exciting technology. It stands for clustered regularly interspaced short palindromic repeats. It's really an interesting thing. I don't want you to memorize what the name stands for, but it's used to selectively modify the DNA of living organisms. And what they actually did was studied yogurt that has naturally occurring CRISPR molecules. And CRISPR acts sort of like a pair of scissors and cuts out the piece of DNA that it doesn't want, and then can repair it and sort of restructure those bases to make it a different way. So it's developing, but it shows promise. For example, CRISPR has been used to cut out the gene responsible for sickle cell anemia and replace it with a gene that codes, that makes a protein similar to fetal hemoglobin instead of that sickle-shaped hemoglobin. They have done that successfully in a number of cases and cured sickle cell disease with it. So beta thalassemia, they can do the same thing. They have seen remarkable improvements. People who were going into crisis several times a year, after 5-6 years, had no symptoms of sickle cell anemia or beta thalassemia after their genes were manipulated. So Victoria Gray, and you can read about this if you want to, pop it into a search engine. She received treatment with CRISPR and has been asymptomatic all these years since. And there have been other cases. The problem is it's really fabulously expensive, and people can't access it if they don't have the funds or the insurance, or they're not part of a research study. They're looking at treatment for HIV and other diseases. They're interested in cures for dementia, Parkinson's, cancer, diabetes. It does have promise. However, there are ethical considerations for CRISPR. People fear CRISPR because theoretically you can edit the human genome in an embryonic cell and make a designer baby. And you're going to watch the video documentary Human Nature, which talks extensively about this. There are issues of informed consent. If somebody manipulates your genome early on, the child, that results from that, the offspring that results, has no way to consent or refuse the alteration in their genome, and they're stuck with it. And whatever is altered will be passed on to their children. Editing the genes of other species, for example, mosquitoes, could lead to unintentional impact on the environment. There's a lot of talk about editing mosquitoes so that they no longer transmit malaria, for example. But when you edit the genome, you might have unintended consequences. And ensuring access to CRISPR-based therapies is challenging. We already have healthcare disparities for treatments that are far cheaper and easily accessible than CRISPR. And when you introduce this technology, who gets to benefit, who does not benefit, that's a huge ethical consideration. Okay, we're going to kind of take a break here, and we'll pick up with the ethical, legal, and financial considerations in genomic-based healthcare.